started a chapter that was plant growth regulators or the phytohormones. So in the series of phytohormones, we studied two major hormones that were auxins and gibberellins. So auxins and gibberellins, two growth promoting hormones. Last class we had studied. Just let's revise and then move forward to cytokinins and other hormones. So as you can see, the plant growth regulators are those chemical compounds that in smaller quantities they affect the metabolic rates and growth patterns in the plant body. So they are found naturally in the plant body, and today they are also synthesized artificially, and the major hormones were auxins, gibberellins, cytokinins, ethylene and abscisic acid. So for auxins we had studied Darwin's and Wendt's experiment, where we saw the phototropic response of the plant tip or the shoot tip was due to the transport of auxin at the shoot tip. So this was auxin experiment or bioassay done by Wendt. So here he took agar blocks rich in auxin and then placed on the decapitated shoot tip, and then he saw the process of bending in the Avena coleoptile. So this is the structure of auxin that we had studied in the last class. The major physiological roles of auxin, where it induced root formation, that is adventitious root formation from the stem cuttings. Then it also promotes apical dominance, that the phenomena of apical dominance was whenever the dominant bud is present at the apex, it is dominant in character, suppresses the activity of the lateral buds. Then it also induced parthenocarpy and also prevents the formation of the abscission zone. Gibberellins were also the hormones that helped in elongation of the internodes of the plants. So here they were discovered from the fungus, the Gibberella fugicori, and this is the structure of gibberellin that we saw, and this is the physiological role of gibberellin. It is mainly elongation of dwarf pea plants, bolting in case of the rosette plant. So this is the phenomena of bolting, where you see internodal elongation leading to flowering in the rosette plants like cabbage and lettuce. And the next was it induced parthenocarpy, it broke the dormancy, and it increased the number of male flowers in the monoecious cucumber plants. It also helped in mobilization of food reserve, stimulating synthesis of amylase enzyme, and then the amylase enzyme was used to break down starch, and then starch broken down into maltose was converted later into glucose, which helped in the germination of seed, or it induced mobilization of food reserve. So in the last class we had studied up to this portion. Today let us discuss the next hormone, the cytokinins. So what are cytokinins? As the name indicates, cyto is a cell, kinins is kinesis or division. So as the name indicates, these are hormones that are going to induce much of cell division in the plant body. So coming to the discovery of cytokinin, the Haberland in 1913, he first reported there are unknown compounds that stimulate the cellular division. So Overbeek in 1940s, he noted that the plant embryos, that is the embryo of the plant, grew much faster when the medium was given rich in nucleic acid, or the coconut milk which is rich in the nucleic acid, it is helping in the division of the cell. So there were two major contributors to the discovery of cytokinin. One is Fox-Coog et al. Or his fellow workers, and Carlos Miller. These two persons actually worked much on cytokinin and discovered the effect of cytokinin and the compound. So what Coog did, he worked mainly on tobacco pith cells. So, in the tobacco pith cells, so in the tobacco pith cells, there Skoog here took a culture medium. So, in the culture medium, there was solidified agar medium, and in the agar medium, there was addition of tobacco pith cells. So, tobacco pith cells, as we know, the pith is the central portion of the stem or the leaf, mainly rich in the parenchyma cells. So, these are parenchyma cells which form the pith of the plant body or the main stem, and he took the tobacco pith cells in the tissue culture experiment, trying to help divide those pith cells, and he added, this is the agar nutrient medium or the solid medium, and in the agar medium he also added the yeast extract. There was addition of yeast extract and coconut milk. So, when the pith cells were provided with yeast extract and coconut milk, in the next day he found that the pith cells rapidly started dividing and they formed a mass of cell. So, first, in the first case, he added auxin. So, when auxin was simply added without any yeast extract or coconut milk, the cells did not divide, but the cells of the tobacco pith, they enlarged. So, as we know, the auxin helps in enlargement of the cells, and here, so the cells only enlarged but did not divide. So, when he added yeast extract and coconut milk, he found the next day the culture, in the culture medium, the tobacco pith cells rapidly divided and formed a mass of cells. So, he was able to find out the cytokinesis activity, but he did not identify any such compound. So, he said there was cell dividing activity in the tobacco pith cells. The second famous experiment was carried out by Miller and co-workers in 1950s, and Miller took the herring sperm DNA. So, what is herring sperm DNA? Herring is a fish. So, the sperm DNA of herring fish was taken, and he took a flask . So, in his experiment, herring took a flask, and in the flask he took the DNA or sperm DNA of herring fish. So, in the sperm DNA of herring fish, he found that overnight in the flask there was rapid division or replication of the DNA to form a large number of copies. So, he found that the old DNA which was added to the mixture, it helped, and he could find out that addition of purine bases of DNA, we know the purine bases of DNA, that is adenine, would cause the cells to divide. So, he found that this base of DNA, that is basically the purine base which is adenine, helped in the division or replication of the herring fish DNA. So, he was able to isolate this compound from the herring sperm DNA, which was later known as the compound kinetin. So, he discovered the kinetin compound, and then they tried to find out kinetin like compounds from the plant body, but kinetin did not naturally occur in the plant body, and they were able to isolate later a kinetin-like compound from the maize kernels, so which was called as zeatin. So, the naturally occurring zeatin was taken out from the maize kernels. Zea maize is your maize or corn. So, the young kernels of maize, from it was extracted the compound zeatin. So, zeatin is also found in the coconut milk. So, all cytokinins, whether they are artificial or natural, they are chemically similar to adenine, that is a purine base. So, this was the discovery of the cytokinins. So, the major cytokinin like compound, cytokinin present in the animal cells is kinetin, whereas the naturally occurring cytokinin from a plant source is zeatin, which is found in the maize kernel or in the coconut milk. So, this is the structure of the common cytokinins. So, this is kinetin. So, this is the purine ring. So, this is the purine ring. So, this is adenine like compound with a purine ring. This is kinetin, and this is again cytokinin of two types. This R, when replaced by this portion, is zeatin, and this is another cytokinin, dihydrozeatin. So, we have zeatin as naturally occurring cytokinin in the plants and kinetin as the cytokinin occurring in the animal cells. Coming to the physiological roles of cytokinin. So, cytokinins have different types of activity. So, cytokinin normally is opposite in activity to auxin. So, it is mainly antagonistic to auxin and works oppositely to auxin. However, cytokinin and auxins are required in specific quantities or ratios to carry out the proper division and elongation of the plant body. So, the first activity of cytokinin is, as the name indicates, we shall see the cell division process. So, it initiates, it helps the cells to divide, and then at the shoot apex as well as the root apex and the leaf. So, as we see that the shoot apex is growing, the leaf is increasing in size, the root apex is also increasing in size or length, that is addition of new cells in these areas of the shoot, root or the leaf apex is by the activity of cytokinin, which helps in the cell division. So, cell division is the major activity of cytokinin. When we use cytokinin in the tissue culture experiments, cytokinins along with auxins play an important role. So, cytokinins promote cell division and in presence of auxin. So, auxin is also required, and in the tissue culture experiment, the balance of these two hormones determine the growth of the root and shoot in the callus. So, what is a callus? The callus is undifferentiated mass of cells. So, the callus is the undifferentiated mass of cell. So, if we are taking this as a culture medium, and in the culture medium we have a few cells taken. So, when we add a specific proportion of cytokinin and auxin in the culture medium, this number of cells that initially was taken as the inoculum, they will start to divide, and this mass of undifferentiated tissue that develops in the nutrient medium is called as the callus. So, callus is undifferentiated mass of tissue that is produced during tissue culture from the cells. So, this callus needs to be again developing into a small plantlet. So, when we are going to differentiate the callus into a small plantlet, we need that there should be production of root and shoot. So, these two types of hormones, that is cytokinin and auxin, here now play an important role. So, the ratio of cytokinin and auxin are important in determining the fate of the callus, what will happen to the callus and what is going to develop from the callus. So, in the callus, if you add a ratio of low cytokinin to auxin, then the callus grows well and it starts forming roots. That means, in this nutrient medium, if I add more amount of auxin greater than cytokinin, then from this callus what is going to develop is, if this is the callus, so this callus will start producing adventitious roots. So, the root system will start getting produced from if auxin is greater than cytokinin. Callus plus high cytokinin. So, taking another activity of cytokinin, cytokinin also helps in the formation of leaves from the nodes. So, what are the nodes? So, if this is a stem, these points having the meristematic tissue from where a branch or a leaf develops are the nodal points of the stem. So, in the nodes which develop into the leaf, cytokinin also helps in production of the leaf. So, this cytokinin gets accumulated at the nodal point and this helps in division of the cell and produce a leaf bud or a shoot bud at the node. So, at the node it will help in the synthesis of or in the production of the leaf. So, the third activity of cytokinin, the cytokinins help in division of the chloroplast. So, one of the major activities again of cytokinin is division of the chloroplast. As we know, chloroplast is a semi-autonomous organelle like the mitochondria inside a plant cell. So, these chloroplasts independently divide, and application of cytokinins over the plant surface help in rapid division of the chloroplast, increasing the number of chloroplast in the leaf or the photosynthetic activity. So, that is another activity. So, it also helps in chloroplast division. So, the next activity of cytokinin in the plant body is an important activity is counteraction of apical dominance. So, apical dominance was a phenomena that we had studied under auxin. That is, whenever the apical bud is present, the concentration of auxin is high and it stops the growth of the lateral buds. So, that was what apical dominance was in auxin. So, here it is antagonistic, that is cytokinin works opposite to auxin and counteraction of apical dominance occurs. So, cytokinin will stimulate the growth of the lateral buds and it will relieve them from apical inhibition. So, this is the apical dominance. So, in this figure, the apical dominant or the apical bud present stops the activity of the lateral buds. If you apply cytokinins, the apical bud, if you remove the apical bud, the lateral buds normally start growing. But even in presence of the apical bud, if you apply a solution of cytokinin, then the lateral buds as here will also start growing even when the apical bud will be present. So, that is where it removes them from apical inhibition. So, this is an important activity and an activity that is opposite to that of auxin. So, going to the next activities of cytokinin, what happens for cytokinin? So, it also helps in breaking of dormancy of the seed and helps in their germination. So, cytokinins are important to break the dormancy of seed and help in their germination. So, we store the seed. So, the seeds are under dormancy or in the resting period. Application of cytokinin will help to break that dormancy and help in division of the cell of the embryo so that the seed starts to germinate. The important function of cytokinin is also the prevention or delay of senescence. So, the prevention or delay of senescence is an important activity again for cytokinin. So, what is senescence? Senescence is the process of aging in the plant body. So, how we know that senescence is occurring in the plant? So, there will be certain symptoms for senescence that we can see. So, there will be rapid disappearance of chlorophyll, degradation of proteins are two important symptoms of senescence. So, commonly what you can observe when senescence is occurring, yellowing of the leaf, so as we see in the plants, there is the leaves start to become yellow. So, it shows the disappearance of chlorophyll. So, chloroplast will no more divide and then the chlorophyll will also start to disappear. So, chlorophyll will be converted into carotenoids. So, that is one symptom where senescence is occurring. There will be also degradation of proteins. You can find that the fruit size, the flower size starts to become smaller and a fewer number of flowers or fruits starts to be produced in the plant body. So, that is the phenomena of senescence or aging in the plant. But cytokinins, application of cytokinins to these plants delay the process of senescence. So, how they are going to delay the process of senescence? Previously, as I have told, that they help in the division of the chloroplast. So, division of the chloroplast is helping to produce new chloroplast, so more of chlorophyll will be synthesized. So, they will delay the process, and this process of delaying the phenomena of senescence was studied by Richmond and Lang in 1957. So, it is commonly also called as the Richmond and Lang effect. So, the Richmond and Lang effect is also used commercially wherever you want to reduce or slow down the process of senescence when you have leafy vegetables or you need to increase the shelf life. So, what is shelf life? Shelf life is how many days you are able to store the product, that is fruits or vegetables or flowers can be stored after taking it out from the plant, that is the shelf life. So, when leafy vegetables are used like your spinach or other leafy vegetables, you can increase the shelf life or the storing period of the leafy vegetable by reducing the process of senescence or avoiding the process of senescence by application of cytokinin. So, cytokinins are applied to leafy vegetables, the green vegetables also to reduce the process of senescence. So, that is the Richmond and Lang effect. The next effect of cytokinin is promotes the production of female flowers. So, it will be promoting the production of female flowers in the plant, and it is applied to keep the harvest products like fruits and vegetables fresh for a longer period, that we now discuss that by reducing senescence or the destruction of chlorophyll and production of more chloroplast, they are used to keep the harvest product like leafy vegetables or the fruits fresh for a longer period. They also are used to keep the cut flowers fresh. So, this is a common activity also we observe, that in the cut flowers which are stored in the flower shop, they also are sprayed with a dilute solution of cytokinin, so that the senescence process is delayed and they remain fresh for a much longer period. So, these are mainly the activities of cytokinins that we can see. So, cytokinins are also helpful in mobilization of nutrients in the plant body. Suppose this is a leaf which is healthy and this leaf is diseased. So, here cytokinin plays an important role here, where the amount of photosynthesis is very less, the amount of food available here is less for the cell. The cytokinins help in mobilizing the nutrients from this leaf to this leaf where nutrient is less. So, the another activity here is also mobilization of nutrients. So, they also help in the mobilization of nutrients in the plant body. So, these are effects of cytokinins in the plant. So, cytokinins are opposite in activity to auxin. They mainly induce cell division. They are opposite in auxin for apical dominance, that is they will be removing apical dominance and they will allow the lateral buds to grow, and so they will also be helping in producing a bushy habit in the plant body. So, then they also delay the process of senescence, which is an important activity for the cytokinins. So, let us come to the next hormone, which is ethylene. So, ethylene here is a gaseous hormone. Ethylene is a gaseous hormone and it is effective in very low concentration. So, how was ethylene actually discovered? So, how did we know that ethylene also plays an important role in the plant growth and development? So, it was found that they found that in the orange orchard, the bananas that were placed were ripening much faster than their normal rates. So, they thought that in the orange orchard or the orange garden, where the oranges were plants were actually releasing some kind of a substance that were allowing the bananas to ripen much faster. Similar activity was also seen in the streets of Germany. So, in the streets of Germany, where the street lamps mainly contained ethylene gas for lighting, they found that the plants there, they started shedding their leaves much faster. That is, senescence was occurring much faster in these trees, and also the ripening of fruits was much faster. So, ultimately they thought there was something in the surrounding that was actually helping or promoting this process, and they later found that it was a gaseous substance, and that is ethylene. So, later then the activity of ethylene was studied in the plant body. So, this is the structure of ethylene that you can see. So, it is a growth regulator here, and ethylene has some positive effects and certain negative effects in the growth of the plant body. So, it can be a growth promoter, at times it is also a growth inhibitor for certain activities in the plant body. So, it is a growth regulator synthesized by most of the plant organ, and it is mainly found in the ripening fruits and aging tissues. So, it is an unsaturated hydrocarbon. You can see it as a double bond having double covalent bonds between adjacent carbon atoms. So, we are all familiar with the structure of ethylene. So, this is the ethylene gas. So, what are the physiological roles of ethylene in the plant body? What does ethylene do in the plant body? So, ethylene here, the major activity of ethylene, coming to the first activity, that is ethylene promotes the ripening of fruits. So, it is commonly called the fruit ripening hormone. So, example in tomato, apple, mango, banana, etc., we find the ripening process is initiated by spraying of the ethylene type of products, which is ethipon. So, here there is faster ripening that you can see in the market for mangoes, half yellowish and half greenish. So, in 1 or 2 days the ripening process is fastened by the application of ethylene. So, ethylene here, what does it do to ripen the fruits? So, as a common experience, we have seen when a group of fruits are kept together and there is one ripened fruit that is present there, slowly it starts promoting the ripening of the other fruits that are kept nearby. And in the process, if we touch these fruits, we find that they are somewhat heated up, or there is release of heat in the particular area which fastens the rate of respiration. So, whenever ethylene gas is released in these fruits during ripening, there is a sudden increase in the rate of respiration in these types of fruits. So, such types of fruits which show a sudden rate of increase in respiration. So, what is respiration? That is, it is breakdown. What is photosynthesis? It is synthesis. So, in respiration, when the fruit that is stored is broken down or converted, so there again heat is released. So, here the heat release that is seen helps in the ripening of the nearby fruits that are present. So, ethylene promotes the ripening of fruits. Such fruits that are able to show a sudden increase in the respiratory rate are called the climactic fruits. So, they are called as the climactic fruits. So, the climactic fruits include the fruits like the bananas, tomatoes, apple and mangoes. So, what is the characteristic feature? What is the importance of such fruits is that we are able to transport, or the transportation of such fruits in the agriculture harvest is always done in the unripe condition or in the green condition. So, whenever these fruits are transported, they are always transported in the unripe condition, so that the damage to the fruits due to over ripening or ripening can be minimized during long distance transport. So, such fruits are called as the climactic fruits. So, ethylene helps in promoting the ripening of the climactic fruits like tomato, apple, mango and banana. So, that is the first important activity of ethylene. So, ethylene here again we take ethylene's triple response as an important factor. So, important character or activity of ethylene is called as the triple response. This triple response is found in the dicot seeds. Can be studied in the dicot seed. What does ethylene do during triple response? During triple response, ethylene can carry out three types of activity. The first activity can be horizontal growth. The first activity that can be seen is horizontal growth. The second activity may be in the seed, which is called appearance of hook. Appearance of hook-like structure. And third response that can be seen is broadening or thickening. Broadening of the shoot. So, this is mainly seen during the germination of the dicot seed. So, what are the three triple response activities that can be seen? One is, if this is the seed, the first can be seen is the plumule starts to grow horizontally instead of elongating upwards. Instead of vertical growth, there can be horizontal growth due to effect of ethylene. Second, this seed plumule can also give you a hook-like appearance. So, there can also be production of an hook-like appearance, or there can also be the increase or broadening of the plumule. So, the shoot can also be broadened. So, you have the triple response activity. It can be horizontal growth, it can be appearance of a hook or the broadening of shoot. So, this type of response of ethylene is commonly called as the triple response, mainly seen in the dicot seeds. So, this is one important activity that you can go through even for ethylene. So, the other activities of ethylene can be, it inhibits the elongation of stem and roots in the dicots, and ethylene hastens or fastens the activity of senescence of leaves and flowers. So, previously we saw cytokinins delayed the process of senescence. Here what is happening? It is fastening or hastening the rate of senescence. So, if there is ethylene, there will be much faster shedding of the leaf that was seen in Germany during the street lamping phenomena. So, it also helps in shedding of the flowers. Many times we see the flowers and leaves fall much faster than their regular time. So, there is senescence. Ethylene stimulates the formation of abscission zone in the leaf, and this leads to premature shedding of the flowers or leaves that is present. So, abscission zone, if this is a stem and the leaf, so commonly whenever there is senescence, there will be yellowing of the leaf, and after yellowing of the leaf, we will see that this leaf is shed. But whenever it is shed, there is no cut mark that we find over the stem, and there is no oozing out of any sap from the cut end or the shed end. So, that is mainly due to the formation of a zone called as the abscission zone at the base of the node in the stem. So, if this is the base of the node, here you can find the cell that are present here will be developing into an abscission zone. In the abscission zone, the cell wall of these cells will be deposited with a suberin like compound or suberin, which stops the connectivity between the leaf and the main stem. So, the vascular connectivity between these two components, that is the leaf and the stem, is stopped when there is formation of an abscission zone. So, this is the abscission zone. So, when the abscission zone is completely formed, this leaf is not getting any more water. So, here it will be shedding off. So, ethylene here promotes the formation of abscission zone. Abscission is important, but ethylene stimulates the formation of abscission, and so there can also be premature fall of the leaf or the flower or the fruit. So, the ethylene is also promoting abscission. So, it promotes abscission, it also promotes senescence, and in the phenomena of senescence, it is also promoting the rapid respiration of the fruits. So, there is ripening of the fruit. Ethylene breaks the dormancy of bud and seeds and storage organs. So, ethylene here will also break the dormancy of the bud, seed and storage organs like your potato tuber or such storage products will be also, the dormancy will be broken by ethylene. So, this is mainly the activity of ethylene, and the another major activity of ethylene is that it promotes horizontal growth or radial growth of the stem. So, ethylene helps to increase the stem diametrically instead of vertical growth. So, that is also an activity of ethylene. So, it promotes horizontal growth. So, horizontal growth is promoted again by ethylene. So, these are the activities that are carried out by ethylene, and coming to the last hormone of discussion is abscisic acid. So, this abscisic acid, commonly known as ABA, is a growth inhibitor hormone. So, all the hormones that are acting in the plant body, that is auxin, gibberellin, cytokinins, are majorly the growth promoting hormones. So, they will increase the height, they will induce cell division. So, production of flowers and fruits and structure, all structures will be produced by mainly the effect of auxin, cytokinin and the gibberellin, internodal elongation and so many activities. But these activities also have to be regulated by a compound, and that compound is abscisic acid. So, abscisic acid is a growth inhibitor which regulates abscission and dormancy. So, abscission and dormancy is mainly regulated by abscisic acid. It increases the tolerance of plants to various kinds of stress. So, when abscisic acid was discovered in 1960s, there were 3 different compounds that were first named. So, one was abscissin, inhibitin and dormin. So, according to the activity of such a compound, later it was found that these 3 structures or these 3 chemicals were same and their composition is abscisic acid. So, abscisic acid is therefore also called a stress hormone and it is mainly produced during stressed environmental conditions where the plant is growing. So, it is also found in the chloroplast of the plant, and this is the basic structure of abscisic acid. The physiological role of abscisic acid. What is the physiological role of abscisic acid? Abscisic acid promotes the process of abscission. Just previously we saw what was abscission. It was the separation of the leaf, flower or fruit from the branch. It is an essential activity because the older leaves have to be shed, and then the same point will start developing a new leaf or a young leaf. So, this is a healthy green plant, and there slowly you see the phenomena of senescence. In senescence there will be yellowing of the leaf or breakdown of the chlorophyll, and ultimately this leaf will be shed. So, how abscission is important? Abscission is important to remove the older and aged leaves from the plant and add the new leaf. So, here this is abscission, this is the new bud that is going to come out, this is the abscised leaf or the leaf that is going to be shed. So, that is one main activity of abscisic acid. Second is during water stress and drought condition, whenever there is less amount of water, we know that abscisic acid causes stomatal closure. So, the closing of the stomata is regulated by abscisic acid in the guard cells. So, abscisic acid starts to close the stomata whenever there is water stress, so as to prevent large amounts of transpiration or loss of water from the plant body. So, it is also called as the stress hormone. The abscisic acid, like ethylene, also promotes senescence and by causing the loss of chlorophyll, and abscisic acid induces bud dormancy. So, during winter when the temperature is much low, there in the plants the buds have to be protected so that they should remain in a dormant condition to overcome the low temperature. So, abscisic acid is synthesized in a larger quantity, and then it induces bud dormancy. Similar dormancy is also induced in the seed. The seeds that we store have large amounts of abscisic acid produced, which prevents them from germination. So, it induces dormancy in the seed as well as buds, and mainly the bud dormancy is seen when there is approach of winter in plants like birch. Abscisic acid is a powerful inhibitor of lateral bud growth and in tomato. So, in tomato, abscisic acid prevents the growth of the lateral buds. Abscisic acid can also cause internodal length in the rice plants that are submerged in water. So, the submerged part of the rice plant in water shows internodal elongation, so that the internode elongates and raises the stem above the water and brings the leaf or petiole also above water when there is flooding in the rice plants that remain submerged in water. So, these are all activities. So, here the abscisic acid is actually regulating the activity of cytokinin, gibberellin and auxin, so that under stress condition or whenever there is need of dormancy or a resting period for the bud or the shoot or the seeds, it can be carried out. So, these were all the 5 different types of plant growth regulators or the phytohormones that we use. The abscisic acid also is commercially used and anti-transpirant, and they are sprayed over the plant in agriculture field, so that the transpiration rate is reduced by causing stomatal closure. So, here we come to the end of the chapter on plant growth regulators and the different topics under plant physiology. So, thank you all for patiently listening to the lecture. Hope you have understood.